Hello, everyone. My name is Aladdin Riad, and I'm currently a postdoctoral research fellow in the lab of Dr. Robert Mack at the University of Pennsylvania. Today, I will be discussing our recent work titled Differential Gene Expression by RNA-seq in sigma-2 receptor TMEM97 knockout cells and what it revealed about its role in complement activation and SARS-CoV-2 viral uptake. So to set the context for our current study, I will begin by giving a little background of our previous work. At the third International Symposium on Sigma receptors, I presented data on the role of TMEM97 in the endocytosis of LDL. In this study, we discovered that TMEM97 forms a complex with PGRMC1 and the LDL receptor, and that this intact trimeric complex facilitated effective uptake of LDL. We identified this complex by studying the protein's interactions and localization using pairwise proximity ligation assays, which are techniques analogous to FRET. In the microscopy images you see here, you will only see a signal if the two proteins being studied are within the length of their antibody pairs, which is about 40 nanometers, and then that effectively indicates that they are forming a complex. The series of experiments indicated that this trimeric complex is necessary for effective uptake of LDL. When TMEM97 levels are low, such as in quiescent tumor cells here, the complex formation is reduced due to those lower levels of TMEM97 and then result in a slower rate of cholesterol uptake. However, in proliferating cells that have a much higher demand for cholesterol, such as tumor cells or metabolically active cells, TMEM97 expression is increased and binds to PGRMC1 and the LDL receptor, resulting in increased rate of LDL uptake. So naturally, our next question was to see if this system existed in other tissues such as the CNS, and if it had a role in the uptake of other lipoproteins such as apolipoprotein E or an intrinsically disordered protein such as amyloid beta 42. Last year, I presented our work looking at the role of TMEM97 in Alzheimer's disease. Our PLA data indicated that the trimeric complex is indeed intact in neuronal cells, and that the disruption of this trimeric complex led to a decrease in the uptake of A-beta 42 monomers and oligomers, the three major isoforms of apolipoprotein, ApoE2, ApoE3, and ApoE4, and A-beta 42 in complex with either ApoE2, ApoE3, or ApoE4. Since TMEM97 was looking like an ideal drug target to inhibit A-beta 42 and ApoE uptake, naturally, our next question was to see what are the genetic consequences when TMEM97 is knocked out. So to answer this question, we utilized RNA-seq. Our workflow was to use our HeLa model system, where we generated TMEM97 knockout cells using CRISPR-Cas9, isolated the RNA, sequenced and quantified, and then analyzed that data. These are the top differentially expressed genes in our TMEM97 cells. On the left are the top 25 downregulated genes in the knockout cells, and then on the right are the top upregulated genes. It was no surprise to us when initially looking at this that the majority of these genes played a role in the molecular pathway modulating cancer development and progression, as TMEM97's role in cancer has been well established and a topic of interest for quite some time. However, what stood out to us from these results were these two genes, C4BP-B and C4BP-A. Both of these genes play a role in inflammation, thrombosis, and coagulation. So to take a deeper look at the function of these proteins, the complement component C4 binding protein consists of alpha chains and a single beta chain that forms this characteristic octopus structure. The main function of this protein is to inactivate the complement system. Here, during the natural turnover of cells upon damage, cells have to be removed from the system. Apoptotic and necrotic cells are recognized by C1q, MBL, and ficolins, which initiate opsonization of the cells. If C4BP is not present, or if C4BP binding fails, as shown on the left side of this figure, the complement cascade is not inhibited and the membrane attack complex, the lytic pore, forms and releases the intracellular content. Professional macrophages, which encounter these opsonized and lyse cells, attack them and release significant amounts of TNF-alpha, thus creating a pro-inflammatory environment. However, if C4BP is present, as shown here on the right, it binds cells and cleaves C3b into inactivated C3b. This allows macrophages to encounter these marked cells and remove them from the system without initiating inflammation. In fact, macrophages with C4BP labeled apoptotic or necrotic cells actually tend to release anti-inflammatory cytokines. Thus, elevated C4BP inactivates the complement system and results in decreased inflammation in the tissue. Another role of C4BP is in modulating coagulation. Protein C is a coagulation factor that binds to cofactors such as the vitamin K-dependent protein S, becoming activated protein C. This activated protein C can then inhibit the formation of thrombin. C4BP binds protein S and can function to modulate this coagulation pathway, which depends on a variety of factors in the local environment. So our next question was to see if TMEM97 ablation resulted in an increase of C4BP secretion at the protein level, or if the regulation was limited to increases at the transcriptional level that we had observed in our RNA-seq data. So to answer this question, we looked at what effect knocking out TMEM97 would have on C4BP protein levels. C4BP is a secreted protein, so we collected media from control cells and from TMEM97 knockout cells and quantified their concentration via ELISA. We found that ablation of TMEM97 results in a significant increase in the secreted C4BP levels compared to control cells, which were TMEM97 competent. Naturally, we wanted to see if we could also cause this increase in C4BP secretion by treating our control cells with specific TMEM97 ligands. We treated our control HeLa cells with RHM4 and observed an increase in C4BP levels in a dose-response manner. At the highest concentrations treated, C4BP secretion was increased to concentrations similar to treatment with IL-6, which we used as a control, as is already known to induce the secretion of C4BP in numerous previous studies. So this was a fascinating finding. We could target TMEM97 to reduce inflammatory effects and modulate the coagulation cascade. So this brought us to our first conclusion that TMEM97 regulates the complement system through C4BP secretion, unraveling a new function for TMEM97 and its role in inflammation. So around the same time that we were conducting these experiments, this paper was published that I'm sure many of us have read. This paper excited a lot of us in the Sigma-2 community. And so what the authors did is they mapped out the SARS-CoV-2 viral, amongst the many things that they did, is they mapped out the SARS-CoV-2 viral proteins and identified which cellular proteins that those viral proteins interacted with. TMEM97 was one of the proteins that interacted with viral protein ORF9C. Sigma 1 and 2 receptors in this paper were identified as druggable targets to inhibit SARS-CoV-2 viral infectivity. So, however, there was one caveat with this study is that they used relatively non-selective Sigma 1 and Sigma 2 receptor compounds. And all in all, this study really excited us and raised our next question was, could we use our model system and our specific compounds to study the cellular uptake of SARS-CoV-2? And by our model system, I mean our HeLa model system where we can knock out TMEM97. So to do this, we utilized a pseudovirus uptake assay, which involved first transducing our control HeLa cells and our TMEM97 knockout HeLa cells with human ACE2, which had a fluorescent tag on it. Then the following day, we treated these ACE2 competent cells with a pseudovirus that expressed the SARS-CoV-2 viral spike protein along with a GFP fluorescent tag. Then we were able to quantify this uptake based off the fluorescent readout. Our first finding was that we observed very low baseline levels of pseudovirus uptake in our non-ACE2 transduced cells, which was expected. ACE2 is a receptor for the spike protein. If you don't have ACE2, you're going to have low levels of uptake. However, when we transduced our control HeLa cells with ACE2, there was a significant increase at the rate of viral particle uptake, which was expected as ACE2 has been characterized as a viral target. So this established our assay, showed that if you take our control HeLa cells that have TMEM97 and add ACE2 into them, you see an uptake of the SARS-CoV-2 spike protein expressing pseudovirus. So our next question was to determine the role of TMEM97 in this uptake process and to see how it affected the viral uptake. So we used our TMEM97 knockout cells, and we found that in non-ACE2 competent cells here on the left, the ones which we had not transduced with ACE2, where when you lack TMEM97, it had a non-significant effect on viral uptake. Again, this makes sense. You don't have ACE2. Whether or not you have TMEM97 isn't going to affect the viral uptake. However, in TMEM97 knockout cells that were transduced with ACE2, we found that this absence of ACE2 resulted in a dramatic decrease in viral uptake, bringing it back down to baseline levels of uptake, which we saw in the control cells that didn't have ACE2. So if you lack TMEM97, you have a significant decrease of SARS-CoV-2 pseudovirus uptake. This observation brought us to the conclusion that viral uptake requires both ACE2 and TMEM97. The absence of either one of these two receptors, viral uptake is significantly reduced. This was very exciting finding right here. TMEM97 appears to play a major role in bringing in the virus into cells. So we wanted to take this a step further and ask, do sigma-2 TMEM97 selective ligands inhibit viral uptake? So for this series of experiments, we used our control cells that expressed TMEM97 and treated them with RHM4 and haloperidol. On the left, you see cells that were not transduced with ACE2. In the absence of ACE2, we see that the compounds had no effect on viral uptake, which is what we expected. Again, if you don't have ACE2, the spike protein is not going to be able to recognize the cells. So whether or not, whether you have RHM4 or haloperidol, it doesn't matter. The virus can't recognize the cells. However, over here on the right, when we transduced the cells with ACE2, our compounds had an effect on the uptake of the pseudovirus. Both RHM4 and haloperidol inhibited the infectivity of the pseudovirus. So the conclusion from these experiments was that sigma-2 selective ligands inhibit viral uptake. RHM4 is a selective ligand for TMEM97, meaning and it doesn't bind anything else, meaning if you can target TMEM97 specifically, you can reduce viral uptake, which is a very exciting finding. So this means that selectively targeting TMEM97 is a potential therapeutic to reduce viral uptake in cells, which could reduce the effective viral load. So we started thinking about back to our previous studies where we observed a reduction in lipoprotein uptake in TMEM97 cells. And in those experiments, we found that TMEM97 interacted directly with the receptor for the lipoprotein, which was the LDL receptor. So we dove deeper into understanding what TMEM97's role was in this process by asking this question. Does TMEM97 form a complex with ACE2 that facilitates viral recognition of the cell and uptake? We hypothesized that it could interact with ACE2 and facilitate SARS-CoV-2 spike proteins recognition and subsequent uptake, just as we observed in the context of LDL, recognizing and being taken up by the LDL receptor. So for this set of experiments, we used our HeLa control cells that are TMEM97 competent, and we transduced them with human ACE2. Then we performed a proximity ligation assay to see if these two proteins interact. Our PLA data indicated that they do in fact interact and form a complex, similar to what we observed occurring with LDL receptor and PGRMC1. So this was one of the first times that it has been observed that TMEM97 interacts with ACE2. And obviously this complex was not present in our control cells that have TMEM97 but lack ACE2. If you lack one of the proteins, you're not going to see an interaction. nor in our HeLa TMEM97 knockout cells that were transduced with ACE2, meaning they don't have TMEM97, but they do have ACE2. Again, if you're lacking one of the proteins, obviously there's no complex. This is a control. So taken together, this has described a new interacting partner for TMEM97, the ACE2 receptor. This also described a new function for TMEM97 in facilitating the uptake of viral particles expressing SARS-CoV-2 spike protein. And so in conclusion here, found that TMEM97 binds ACE2, interacts and facilitates with viral uptake within cells, and then upon the loss of or inhibition of TMEM97, viral particle uptake is attenuated. Future studies will determine if this uptake attenuation is due to the binding of the viral particle and subsequent proteolytic activity of TMPRSS2, or if the spike of the spike protein, or if its effect is solely further upstream via ACE2 recognition of the spike protein. Furthermore, in the study, we found that the inhibition of TMEM97 resulted in increased C4BP secretion, which regulates complement activation and is a modulator for the thrombotic effects that have been seen with COVID-19. So to summarize, knockdown or pharmacological inhibition of TMEM97 reduces SARS-CoV-2 pseudovirus uptake. TMEM97 formed a complex with ACE2, and TMEM97 knockout modulates the complement system by increasing C4BP expression. So we started these studies by asking, how does TMEM97 regulation of C4BP relate with COVID-19? And it does so by modulating some clotting issues that increase the probability of stroke that has been seen in patients with COVID-19. And it modulates the inflammatory response, which again could play a role in, could be therapeutic value, such as in patients that have this intense and inflammatory response to cytokine storm. TMEM97 potentially plays a key role in two features of COVID-19. It mediates cellular uptake of virus. The loss or inhibition of TMEM97 reduces viral particle uptake. And it potentially can modulate systems of COVID-19, including inflammatory response and thrombosis via C4BP regulation. Although vaccines have been developed against the current SARS-CoV-2 virus and some of the new mutations that have been discovered, the virus is constantly mutating, and there may be new coronaviruses as well in the future that current vaccines may not be effective against. So having therapeutics that target cellular machinery would be incredibly useful to limit viral uptake of future viruses and reduce the effective viral load. TMEM97 may potentially be a therapeutic target that curbs the rapid spread of future viruses and limit the effect it has on the population until future vaccines can be developed. With that being said, I'd like to thank my mentor, Bob. I'd like to thank my lab members, especially Tom Graham, who spent a lot of time discussing these ideas for this project with Bob and me. I'd also like to thank our collaborators, Brian and Jan, who helped us out tremendously with analyzing the RNA-seq data, and our collaborator, James Peterson. I'd like to thank the University of Pennsylvania Department of Radiology, and I'd like to thank everybody here at the International Symposium and Cognition for putting this on. Thank you very much, and I look forward to our discussion sessions.